Assalamualaikum and hello everyone. We are from Group 13 and we will present about cell death. My name is Nadia Nabila and my metric number is 208304.
My name is Chuan Xiaoyi. My matrix number is 209131.
Hi everyone, my name is Nur Qurratu Shakila binti Zulfaqar. My matrix number is 207871.
My name is Nur Tanya binti Kamar Zaman. My matric number is 206824.
Okay everyone, please enjoy our video.
Hi everyone, my name is Shakila and today my group and I will be presenting on the topic cell death. So what cell death is actually about? Cell death can be described as when a biological cell terminates its normal cellular activities. Cell death usually happens when there are injury, infection, and death of the organism. Cell death also said to be critical as cell death controls cell number for normal development, defensive strategy from unwanted cell, and maintains homeostasis. For instance, P53 is a tumor suppressor gene that is part of the cell cycle control machinery known to induce cell death. P53 responds to DNA damage and stress condition by activating transcription of genes. Moving on, there are three main mechanisms of cell death, which are necrosis, necroptosis, and apoptosis. So we have necrosis, which is triggered by acute injury or insult, like trauma, and lack of blood supply. While necroptosis is a type of programmed cell death triggered by signal from the other cells. In both necrosis and necroptosis, cell bursts and contents release. This may then trigger inflammatory response.
And I'm going to talk about apoptosis, a type of mechanism of cell death. Apoptosis is different compared to necroptosis and necrosis because the cell will not burst and it will not trigger inflammatory response. So let's proceed to see what's the main characteristics in apoptosis. The first notable characteristic is there are blebs on the surface of the cells. Then the second characteristic is the breakdown of the cytoskeleton in the cell. The third one is the breakdown of the nuclear envelope. The following one is the condensation of chromatin in the nucleus. After the condensation of chromatin, it will proceed to the DNA fragmentation process. After that, the cell will start to shrink and it will form a few apoptotic bodies. Then the phagocytic cell will engulf all these apoptotic bodies. Then it can be reused by the phagocytic cell itself or the other cells. Here's an example where the apoptosis will take place. It is when there is accumulation of misfolded proteins in the rough endoplasmic reticulum which trigger the unfolded protein response UPR. It will signal the rough endoplasmic reticulum to expand its capacity to increase the synthesis of proteins, which is important for the quality control in the ER lumen. For example, chaperone proteins. If the endoplasmic reticulum has reached its maximum capacity and the cell is still unable to cope with the situation, apoptosis will be triggered. So here's the overall of the apoptosis cycle where the cell will become apoptotic bodies and the apoptotic bodies will be engulfed by the phagocytic cells. Then the content can be reused by the phagocytic cells itself or the other cells.
Now, I am going to explain on how does the apoptosis can be triggered to take place in a cell. So apoptosis is mediated by caspases in an intracellular reaction called the caspase cascade. First of all, what is a caspase? Caspases stand for cysteine-dependent aspartate-directed proteases. They are a type of protease enzyme which serve to break down proteins. There are two types of caspases involved in apoptosis, namely initiator caspases and executioner caspases. Initiator caspase, for instance, caspase 2, 8, 9, and 10, is firstly made as an inactive precursor and is known as an initiator procaspase. It has an adapter binding domain and a protease domain which contains a cleavage site. In the caspase cascade reaction, the initiator procaspase firstly needs an activation in order to become an active initiator caspase. The activation can be induced in response to apoptotic signals. When there are apoptotic stimuli, such as unneeded immune cells, cells with damaged organelles, or cancer cells, these apoptotic stimuli will trigger the assembly of adaptor proteins. The assembly of adapter proteins will promote the dimerization of the initiator procaspase monomers. They do so by bringing together a pair of initiator procaspases and bind to their adapter binding domains. This action activates the initiator procaspase, which then leads to the cleavage of their cleavage sites and become an active initiator caspase. Once the initiator caspase is activated, it will then activate downstream executioner caspases.
Now, we will move to the next part. Generally, executioner procaspase is an inactive form of executioner caspase which will carry out the mass proteolysis that leads to apoptosis. In order to understand more about this topic, we need to capture the key point first. Now, let's do this together. The first key point is executioner procaspase initially form as inactive dimers which contain large and small subunits and cleavage site. Next, active initiator caspase which take part during the activating process will cleave the executioner procaspase. The active form is called executioner caspase. Furthermore, after the cleavage, the executioner procaspase will undergo an activating conformational change in order to be active executioner caspase as shown in the diagram. Last but not least, executioner caspase will trigger apoptosis by cleave variety of key cellular proteins such as nuclear proteins, plasma membrane proteins, and mitochondrial proteins for the cell to dismantle itself quickly and neatly. In summary, executioner caspase such as caspase 3, 6, and 7 are activated by cleavage between the large and small subunits and are therefore dependent on initiator caspases for their activation. The cleavage of caspase substrates can result in loss of function of target proteins and eventually leads to the cellular change associated with apoptosis. Now, we will go to the intracellular proteolytic cascade, which is a series of reactions that is irreversible when it started to act like a very destructive process and makes the apoptosis need to be tightly controlled. It will amplify the chain reaction when the initial activation of a small number of initiator caspases can lead to the explosive activation of a large number of procaspase molecules to cleave a number of key proteins in the cell that will lead to apoptosis at the same time. I think that's all from us, and we hope that you guys enjoy this video, and don't forget to like and share this video with your lovely